Typically, first in human studies are performed with a simple fit for purpose formulation. That's usually a drug and powder and bottle or capsule, something very quick and easy. However, those product formats are unlikely to be suitable for phase 2 studies, potentially when you start increasing your patient sizes to 100 or more patients and having multiple sites with longer durations for dosing. Most early development programs therefore require management of formulation change between that first in human studies and your phase 2. The three common strategies employed in industry today, you can see here in the middle. The top one there is the probably the most simple, most direct, which is a liquid-based system for delivery. We meet that requirement for a rapid initiation for a first in human study by utilizing a simple solution or suspension. And you can get data on safety with minimal CMC investment while still getting, your tolerability, PK exposure. However, for the drug product program itself, you're really pushing the development of the program just down the line. So therefore, there could be a slight delay while you do some additional development after your phase 1 and bridge those formulations for your patient studies. Second strategy we can employ is to start the first in human study with the long-term goal in mind. You can develop a solid dosage formulation, which could have legs that possibly be pushed into phase 2 that's suitable for those patient studies. However, in the absence of any existing human data, there are some inherent risks and uncertainties with taking that approach. There's also quite a bit of CMC investment at the front end for this approach prior to initiating any studies in your first in human. And while there could be some potential timelines or impact on the front end, there's also some potential time and impacts on the downside of this after you come out of your first in human, particularly if you're not getting the desired results and exposure that you're looking for. The third option that you see in front of you here is a hybrid strategy. So you can break into your first in human protocol with a bridging study, for example, between a SAD and MAD study, and you can develop models to practice that cost and time savings to gear to the needs of today's development programs and make it a little more simple and patient-ready formulation and still viable for something to be carried on into your patient studies.